Good morning, good afternoon, or good evening, depending on where you are in the world, and welcome to another day of MDDI's Focus on Fundamentals, Big Changes in Medical Device Biocompatibility, sponsored by Nelson Labs and broadcast by Informa. It's day 2, and we'll be discussing updates to USP Class VI impacting medical device materials. I'm Omar Ford, and I'll be your moderator today. We have just a few announcements before we begin. This webinar is designed to be interactive. The dock of widgets at the bottom of your screen will allow you to learn about today's speaker, download resources, and participate in the Q&A session that takes place at the end of our presentation. The slides will advance automatically throughout the event. Also, if you haven't already, be sure to click the Register Now button in your viewing window and join us for the remaining Focus on Fundamentals class tomorrow, where we'll be discussing how to address updates for existing products and in-process submissions. At the close of tomorrow's presentation, we'll wrap up with a panel of all of our series experts addressing your questions. Lastly, if you're experiencing any technical problems, please click the Help widget found at the bottom of your screen, or type your issue into the Q&A area, and we will be glad to offer one-on-one assistance. Now, on to the Day 2 presentation, updates to USP Class VI impacting medical device materials. Discussing today's topic is Nicholas Cristiano, Global Market Segment Leader, Biological Safety at Nelson Labs. To learn about our speaker, please visit the speaker widget. Nicholas, over to you.
Thank you, Omar. As mentioned, today we'll be discussing timely changes to USP Class VI updates, including USP Chapters 87 and 88, including additional informative chapters as well, where we'll focus on the shift from traditional Class VI testing to more of a risk-based approach aligned with ISO 10993. We'll explore a little bit of history, problems with past approaches, and some new in vitro alternatives. So getting started, a little bit of history about the USP and what got us here and kind of where we are today. The USP started back in 1820 when a group of 11 physicians met to the US Senate, basically around their concerns about poor quality of medicines and how they're prepared. Congress had then, moving forward, passed a Pure Food and Drug Act, which gave legal authority to the standard. That really drove a lot of the construction of the USP chapters from there. So the initial, or the current USP mission statement, that is to improve global health through public standards and related programs to help ensure the quality, safety, and benefit of medicine and foods. USP is also largely a voluntary program. And so whenever we do discuss changes in USP, we do want to be aware that there is a lot of work going into these documents that is voluntary from industry, testing facilities, CROs, and those who really want to move industry forward in these standard documentation. So really always kind of nice to keep that in mind, and we like to thank those involved in that documentation. So like I mentioned today, we will discuss both informative chapters and the other chapters, the application chapters, the USP, that are associated with Class VI material testing. Currently, combination products are expanded upon the scope of USP to include medical device materials as well. So something to remember there is that finished final medical devices are still subject to their own regulations. I think we all probably are well aware that our finished final good is really governed by ISO 10993, and that something to keep in mind as we talk today is that USP will be more relevant to our materials manufacturer. So just a few other points as well as we move forward. Back in 1975, that is where we see USP Chapter 88, which are our biological reactivity tests. So those are our in vivo tests, our animal model tests. And then about 15 years later, in 1990, we saw the addition of Chapter 87. So not to be too confusing, but to kind of go back a number there, where we titled that chapter Biological Reactivity Tests in Vitro. So these are where we see our cellular assays that we'll discuss here shortly as well. Okay, and as I mentioned, we'll start with talking a little bit about class designation and chapter review of chapters 87 and 88. So what are classes? Key points about each class really we'll discuss in a following slide where we can understand a little bit more. But the main really idea here, the main concept was that as we increase risk in materials, we increase the classification, and then we therefore see the number of tests required for those classifications increase as well. So simply put, class 1 is the simplest material. Class 6 would be the most comprehensive. And as I mentioned, we also see an increase in number of extraction solvents, typically with each class identification as well. So following that nomenclature, higher classes would use more extraction vehicles to simulate those different chemical polarities and so on. Implantation testing, we typically have seen starting around the class 4 as well. And so this is to observe tissue reactivity directly at the implant site. We'll talk a little bit more about why that test has seemed pretty irrelevant in historical use, given the fact that we are testing materials and not finished goods, and also typically materials that are not implanted. But what we did find out is over years, class 6 really became the industry default. So even though it was designed for, as I mentioned, raw material qualification, one, for pharma containers and pharmaceutical products, not quite finished medical devices, the industry tend to kind of overuse it because of how risk adverse the medical device industry tends to be, right? So we tend to always assume the worst and say, maybe we should test 2 class 6 to be able to market a little bit more appropriately. So we'll talk a little bit more about that later on as well. And really, historically, we've seen an animal-based approach here. So similarly to other standards, in the medical device area and also in pharmaceutical container enclosures, a lot of use of animal models historically were most prevalent. So in this historical case, all of our classes, as we're about to see broken down, relied on some form of in vivo tests. And I recognize that this chart may be small, but this is just the historical system classification table, so I wouldn't worry too much about being able to see everything here. I just wanted to use this source reference from USP to kind of explain a little bit more about how that risk-based approach kind of went wrong. So again, initially risk-based, but became quickly overused. This was really, classifications were really applied more to marketing materials to manufacturers rather than to risk relevance. So again, we saw manufacturers really defaulting to USP Class 6 rather than properly identifying individual classes and then leading to less animal use, less testing, et cetera. And so, of course, that resulted then in unnecessary animal testing, which we've seen. And really another driver that really drove, a lot of Class 6 use was the low cost to the testing associated with Class 6. So as a material manufacturer, it was a bit more advantageous for me just to spend a little bit more money, maybe $6,000, $7,000 in today's testing to get that Class 6 designation, if you will. So that kind of encouraged that further default in use as well. Okay, so here we talk a little bit more about the classes from that historic system. So what I've done here is I've just expanded upon that chart that we saw on the previous slide. Again, wouldn't worry too much or get too hung up here. This is the historic system, and we will talking about how we're moving forward. But just to kind of give us a little bit more of a foundation to understand where we started and where we're coming from. As you see on the left, we move down from Class 1 to Class 6. As we move over across to the right of the slide, our tests required become a little bit more demanding, right? So we start with a systemic injection test, and we kind of increase our solvents there. We add a implantation test around class 4, which carries through class 6. And then we move up again, as I mentioned, into additional solvents to reach that highest level of stringency, if you will. So again, you can see here that there was enough of a request for class 6 testing to make sense for material manufacturers because it was a bit more achievable. Okay, so now we're going to start to talk a little bit about changes to each of these chapters. So again, we have, chapter 88 is what we're talking about first, right? Those are biological reactivity tests in vivo. So those are animal models. So here we'll discuss just a few differences between the historic version, which is in the middle of your slide, and our current version, which is on the right-hand side of our slide. So I will mention this a few times throughout the presentation just so that we do recall and we do remember this important piece. But this chapter will go into effect December 1st of 2026. However, the USP is allowing for us to honor this now, effective immediately. So again, just a good chance for us to start leveraging the new changes here. So as we look at the classification scheme, we talked about the 6 numbers of classes 1 through 6. Moving forward, there will actually be no class designation. There'll be new terminology, which we'll discuss a little bit further on. And the reason doing that is we want to try to shift our focus on those relevant endpoints. We want to be a little bit more risk-based. So again, aligning further with ISO 10993, where we see ISO 10993 aligning more with 14971 or risk management standards. So testing, we will talk about this further as well, but we started, with systemic injection, adding in some intracutaneous irritation tests, some implantation tests. Now moving forward, we will only see systemic injection, that in vivo test in particular cases. So we'll talk about that some more as well. Again, so here you see I have listed high risk cases as part of supporting toxicological risk assessments potentially as well. Extraction solvents, again, that kind of follows the test used. So we went from a whole bolus options of extraction solvents down to moving forward, being a little bit more streamlined. Implantation studies have been fully removed. And so we no longer consider those scientifically valid or ethical. We'll talk about that a little bit more. And same in intracutaneous irritation testing. So the animal model has been removed and be replaced by an in vitro model, which will lead us to obviously further discussion in chapter 87, where we'll talk about our tests in vitro. So again, the purpose here of this test prior was a tiered system to screen for those plastics, raw plastics, polymeric materials. And now moving forward, we will be focusing more on those in vitro and chemical characterization methods where applicable. And we will only focus on in vivo models when scientifically justified. So historically, you could think of chapter 88 as being used more widely because we were really kind of using these animal models as a crutch. Now we will be shifting more towards a use of understanding chapter 87 and come back to chapter 88 if needed, if applicable during high risk materials or if needed. And so again, we talked about animal use being significantly reduced. And from a regulatory perspective, these are just supportive tests, as I just mentioned. Okay, so further changes to USP Chapter 88, a little bit more of an easier summary slide here to digest. Systemic injection is a test where we typically see injection either by IV or IP, usually with mice, and then cage side observations. So we're looking for weight changes, behavioral changes when compared to a control. And so historically, that's what this chapter had included, right? Test by systemic injection, also intracutaneous irritation test. That was where we dosed animals with material extracts within the skin of rabbits. We observed them for 72 hours, and we really just looked for redness and swelling, right? So a very qualitative assay here. And now, additionally, as we talked about, implantation was also included. And this was a 5-day implantation, typically in the muscle or subcutaneous in rats, where histopathology was macroscopic. We're looking for things like necrosis, tissue discoloration, immune system responses. And so, one of the reasons why this was considered scientifically unjustified is that most of these materials, as I mentioned, would never really be considered for implantation, and also such a short duration, right? So I think a lot of us here are probably thinking, wow, 5 days, there's probably not even enough time consideration for healing. And so those macroscopic histopathology observations just very, very much seem to be obsolete. And so as we move into our new version of Chapter 88, again, it becomes official in December 2026, but has been published early for early adoption. Now only includes systemic injection. So the test we just talked about, but just used for higher risk materials. And we'll talk about a flow chart about when you would consider that further on here. We remove irritation, we remove implantation. And again, systemic injection can be used to support Chapter 1663, which is our chapter for extractables for understanding our polymer products. Okay, now we move into Chapter 87. And so these are our biological reactivity tests in vitro. So here's where we see a lot of change for the addition, right? So we removed a lot in Chapter 88. We wanted to get away from those animal models, a lot of them that were not predictive, not sensitive, not specific to this use case. And we really want to kind of really buff up Chapter 87 with supporting tools in the in vitro space. So primarily, the old Chapter 87 really just listed cytotoxicity. Now we've expanded to multiple in vitro endpoints beyond cytotoxicity, which we'll talk about here. With that being said, historically, we just saw MEM elution as a qualitative test, agar diffusion as a qualitative test, and then also direct contact. And so moving forward, we do highlight MEM elution still. However, we've added the neutral red uptake, which we'll talk about briefly, as a quantitative cytotoxicity test. And then we've also added in vitro irritation and our in vitro genotox tests. We won't spend a ton of time today on the genotox tests. Those are historic, and we have been working with those for quite some time. We can always go into those further in depth later if need be through discussion with Nelson Labs. But we will talk a little bit about in vitro irritation and our differences between qualitative and quantitative cytotoxicity, so we understand why we made those changes. Okay, now as we move forward, again, quantitative toxicity, it was once not required. Now we do have an option available to be quantitative, and we'll discuss why. Irritation and genotoxicity, as I mentioned, previously not included, now we've added. And regulatory alignment here was really for stronger alignment with the 10993 standard. So Part 5 for cytotoxicity and Part 23 for irritation. Animal use here, obviously we have none. We are talking about our in vitro chapter, right? So intended uses, again, additional screening methods to understand our materials and extracts. And we've expanded the scope here, right? So we've added some quantitative capability, increased a bit more predictive value, and obviously for clinical safety as well. So again, just an easier way to highlight those summaries. Chapter 87, in vitro tests, the current revision only listed our MEM elution, agar diffusion, and direct contact methods. The new reversion, again, effective December of 2026, but available for early adoption now, removes agar diffusion, as most don't believe this test provides much relevant data for what we're doing. We do add the neutral red uptake, which is a quantitative test, and we're adding in vitro irritation. We'll talk about that model here shortly. And we add our 4 genotoxicity tests as well. All right, now we will go into biological testing review. So as we've kind of navigated both chapters 88 and 87 to talk a little bit about changes from the in vivo models and in vitro models recommended by the standard and listed by the standard, we'll talk a little bit about what that biological testing looks like as we kind of move forward here. One thing to kind of remember is we have shifted in really all standard approaches across medical device and pharmaceutical industries, albeit the medical device industry seems to be last to the kind of finish line here, as we are still far from the finish line. But we are moving towards the case for more alternative methods. So we obviously all want to embrace the 3 Rs, which I think we all know are reduce, refine, and replace current methods and animal models to increase animal welfare, provide sustainability in testing, and also provide more sensitive and specific assays that are more relevant to our products. These methods are also more repeatable, quantitative, often less subjective. Cosmetics adopted them pretty quickly due to consumer demand. And so we do find that, we as the customers here, as the medical device companies, and, our customers being our patients and end users, really need to be continuing to drive the demand for the replacement of these models and the refinement of these models to become less dependent on animals and more dependent on sensitive and specific in vitro and in silico models as we move forward. Chemical characterization now we see complements biological testing very well. We've seen that with the introduction and the further push for chemical characterization testing through ISO 10993 and other standards. And so it's great that we're able to continue to do that and move forward. So a few other points on here too is just that we want to, make sure that we are clear that Nelson scientists are very active in validating these in vitro alternatives, have spent a lot of time supporting in vitro irritation models, and now looking forward to further support in the sensitization assays as we kind of move down those pathways. And we do recognize that this change does take time. So again, we need our manufacturers, and our customers and the testing industry to be supportive of this and help us continue to drive change. So brief overview of cytotoxicity. What is cytotoxicity? So this would be cell death from toxic substances, right? The presence of something toxic in that cellular environment. These requirements within the USP chapters are harmonized with ISO 10993 Part 5. And so typically you may even see these cited as such from your laboratory reports in both ISO and USP citations. We're looking at really here a basic cell culture. It's highly sensitive. It's a great screening tool. We're culturing cells, we're growing them, we're exposing them to extract fluid, and we're determining viability. And again, we use this because it's highly sensitive and a great screening tool for materials, both in pharmaceutical and in medical device industries. So quickly, qualitative versus quantitative, since we will be having these discussions between these different assays here. Really qualitative, the way I like to think about it is what kind of damage happened? How bad does it look? Quantitative, exactly what's going on and what has survived, what is viable, right? And so as we kind of move through here, we'll discuss both those quantitative and qualitative assays and again, why they're important. So qualitative assays, typically when people say qualitative, the first thought goes to MEM elution. This is where we're observing cells under a microscope. We're assigning a cytotoxic grade 0 to 4. And that grade is basically an estimation of the percentage of cell death from what we're seeing on in those images from the microscope. So again, most people refer to this as MEM elution. There's really great advantages here. Qualitative method has a long history of use in the US. It's pretty inexpensive. It's relatively quick. Laboratories are constantly working on developing their cell lines and keeping their cell lines happy and healthy, so they're ready for introduction to these medical device extracts, and these tests can start relatively quick. We can look a little bit about cell morphology and help understanding the difference between what we refer to as cytotoxic and cytostatic. Sometimes that is often a question. So cytotoxic would be toxicity causing cell death. Cytostatic would be slow cell growth. And so sometimes there is an impact for materials there. So we want to make sure that we understand the difference. Again, talked a little bit about primary uses and why there are strong advantages to the qualitative evaluations. Typically, lot release testing, we see this often with supporting cleaning validations. And really, again, just a really good screening tool. I've got a change, maybe a small change. Let's make sure that small change doesn't have any downstream cytotoxic effects. And now quantitative evaluations. So this would be here on the screen. I talk about the MTT assay, but also this does include the neutral red uptake as well, where the neutral red uptake would be where we introduce neutral red dye to our cells. Viable cells would then uptake that dye, and then you would rinse our cells to remove any excess dye, and we would lyse them to release the red color, and we would read that red color in a spectrophotometer. Simply put, the more intense the red, the more viable the culture is. And this happens very similarly with the MTT assay as well. Advantages here are that the method is not subjective. Results are presented by a percentage of viability. And so we don't have any of that subjective thought or nature to the test. We are able to include dilutions of the test extract, which you can do in an MEM elution as well. Quantitative data also aligns well with our toxicological risk assessments often. This is typically the preferred method in the EU. Qualitative typically is also followed by these quantitative assays are followed up by qualitative failure investigations. So ISO actually tells us that if we fail a qualitative method, an MEM elution, for example, you may want to follow that up with a quantitative evaluation. And so that's often where we typically see them used as well. Okay, now a little bit into the in vitro irritation models. So we talked about the replacement of the animal model, the intracutaneous irritation, with what we refer to as a reconstructed human epidermis model. So these two top pictures represent about over 3 decades of research going from these in vivo models into the in vitro tissue. And so you can see both of them listed look very similar. As you can see, the layers of the differentiated cells is present in the in vivo model on the right, similar to that of the in vivo tissue on the left. These tissues are grown from human-derived epidermal cells. Both adult and infant tissues are used. Typically, infant tissues are used of the two different large manufacturers. No differences have really been seen between use of the two. The test is really good for not just skin contacting devices, which is typically where questions come from. I don't have a skin contact device. Should I use the reconstructed human epidermis model? And the reason being is that if we actually look at the stratum corneum on the in vitro model, you can actually see a little bit of white there. You can see a little bit of space. And the stratum corneum in the in vitro test is really great for allowing the passage of bigger chemicals. And so we're able to leverage that for really all device types that require irritation testing as well, and has been validated extremely well. So a little bit more about that model and how it works. This is what we call a 3D tissue construct. And so these are laboratory grown 3-dimensional models, right, of human skin. And these are made from human cultured skin cells. These cells differentiate and organize into the stratum corneum, or the outer layer, as I mentioned before, and viable epidermal layers. What they do is they're mimicking the structure and function of human skin. And so think of this model as a 2-wall container, typically produced on a porous membrane or an insert to support and allow nutrient diffusion from below. So I understand the photo may be a little bit obscure here, but you can see those arrows coming up, so you can see the nutrients feeding the tissue from below through that microporous membrane. And so how do we score this test? So, we're using, again, we're measuring cell viability exposed to an MTT assay. And so this is a quantitative method where colorimetric assay is used to measure the metabolic activity of those living cells. So the test relies on the ability of viable cells, mitochondrial enzymes, to reduce the yellow MTT reagent into a purple agent, or we call them purple formazine crystals. And so the more intense the purple color, the more viable your cells are. So the extract results are compared to a tissue control, which is typically just your solvent in this assay. After exposure to the test substance, cell viability is measured using that MTT assay, as I mentioned in the previous slide. And what's nice about this test is it's essentially a pass-fail. So greater than 50% viability is classified as a non-irritant, and less than 50% viability is classified as an irritant. And so here on the screen, again, I know this bar chart may be quite small, but we can see on the right the positive control looks like it's around 5% or so viability, whereas our solvent controls, those would be oil, saline, listed on the right, are near 100%. Okay, so as we move through chapters 88 and 87, we will start to discuss a little bit more about USP Chapter 1031. And so this chapter focuses on the biocompatibility of pharmaceutical packaging systems and their materials of construction. Listed here by USP states that it's not a mandatory monograph, but it does provide a framework for assessing biocompatibility, again, per the title of the chapter, of polymeric pharmaceutical packaging systems and their materials. And so what that means is, again, further review from earlier, we have informative chapters, which tell us a little bit about how to use. And then we have application chapters, which tell us which tests to conduct, right? So we talked a little bit about that in chapters 87 and 88. And now we're talking about this informative chapter, USP 1031, and the changes here. One really important aspect here is that this new revision expands and modernizes the chapter to better support those risk-based, science-driven evaluations, and again, harmonize the earlier USP chapters we talked about with the ISO 10993 concepts and the direction that that standard has taken. Some practical implications that we see is that there's no automatic grandfathering from USP Class VI. So everybody who has that question saved up for the Q&A, I'll answer it right now. I have materials that have been conducted to historical USP Class VI testing and requirements. But what we do know is that materials must be reevaluated under these new criteria to claim what we used to call USP Class VI. Now we're calling pharmaceutical grade polymeric packaging material. So I'll mention that a couple times during the rest of the presentation, but that's what our new designation is moving forward. 1031 also encourages sourcing well-documented compliant materials from suppliers to help provide a more streamlined risk assessment. We'll talk a little bit more about that. For medical devices, 1031 can help support material selection justifications, but final finished devices still do require an ISO 10993 compliant evaluation, as we briefly discussed. So again, the scope here presents a framework for evaluation of those materials and some guidance on how to support that moving forward. Okay, so now these are two important flowcharts we'll go through briefly. I do recommend reviewing these a little bit further in depth. But what determines testing that's needed now, right? So by this informative chapter to say, what do I need to do to understand my materials and to try to designate the term pharmaceutical grade polymeric packaging material, which again replaces the classification of classes 1 through 6. And we look at this flowchart. We have a risk-based framework. This introduces decision-making for both low risk, like the one we see here, and then high-risk materials, which we'll see shortly in the following slide, emphasizing prior knowledge on materials, compliance statements, and chemical safety assessments before biological testing. So now as we kind of look at the flowchart, we see we really start asking ourselves, do we have compounds of known concern or potential toxicological concerns? If we say yes, obviously we do not have a material that is justifiable. If we say no, we go to our first real decision tree. And so this is conducting cytotoxicity tests and our skin irritation tests. If those pass, then no more testing is necessary, right? Our pharmaceutical grade plastic packaging and material classification can be leveraged. If those tests do not pass, we would start to look at chemical characterization, utilizing USP Chapter 1663. with an individual chemical toxicological risk assessment or a chemical safety assessment, or a combination of those two, which we'll talk briefly about momentarily. If needed, our systemic injection test would then be leveraged. So as we talked about earlier in our in vivo chapter, we do still have our systemic injection test listed, but we are going to try to really postpone using that test until it's absolutely necessary. And if all passes there, then we can apply our new terminology to our material. And now for our high-risk materials, we do have a slightly different flow chart. We're trying to understand, first, is this a solid, right? Is this a powder? Is this, a liquid? We may be able to have a different workflow in those instances. But here we have our flow chart for those high-risk materials, which USP states should be considered, first off, consider being replaced if possible to avoid additional testing, which I think is kind of funny in itself, because if we didn't have that material, if we didn't need that material in the first place, we probably wouldn't be using it. But they do mention that you first want to consider replacing the material for a low-risk material. And if you cannot, here is basically our workflow for additional testing that may be needed, such as analytical chemistry or those genotoxicity tests. So high-risk materials, we have these kind of highlighted on our slide. Think of novel chemistries. We don't have a lot of use case, not a lot of literature across pharmaceutical, or in some cases our medical device applications, a high amount of extractables, toxicological red flags, right, which are appropriate biological or toxicological assessor can help us understand if those are present. Those would require expanded chemical characterization. So we mentioned that USP class, USP chapter, potential toxicological risk assessments, and then also that systemic injection testing, as we mentioned as well. So we do talk about compliance statements, we do talk about chemical safety assessment as a structured toxicological risk evaluation for those high-risk materials to be needed. And they may help you mitigate from a particular material or mitigate the risks from those particular materials. And so as we do that, as we look into these chemical safety assessments, these often include compliance statements from suppliers, information supporting composition, such as base polymers, additives, colorants, what may be included, and basically giving us a materials risk assessment. So in the updated 1031 chapter here, that chemical safety assessment is always done before deciding if any in vivo or in vitro testing is needed. And so again, for those high-risk materials, we want to make sure we're conducting that. And it's also central to both low and high-risk evaluation tasks. So even though it's required and prevalent in high-risk materials, we want to make sure there's a presence of this in our low-risk materials. And so really the goal here is to justify our material safety without any unnecessary biological testing where possible. All right, now we're going to briefly go through chemical safety assessment steps. This is outlined a little bit further, but I want to be aware of leaving enough time for a Q&A session. So we will go through these relatively quickly. But why is a chemical safety assessment important? Again, it allows safety to be justified with using either existing chemical and toxicological data or conducting a little bit of chemical data and avoiding unnecessary animal tests, right? And so we have limited our animal testing now moving forward. However, we do still have some of it relevant within the chapters. It does help us further align USP 1031, again, this informative chapter, with ISO 10993 Part 17 and Part 18. So that's really nice as well. And it facilitates global regulatory acceptance by providing a more transparent, science-based risk argument. So we'll go through these steps here briefly. So step one, gathering material and useful information. So again, as I mentioned, that may be base polymer formulation, any known additives, coatings, I think we're all aware that many colorants contain heavy metal constituents, so we could start to really already understand what our risks may be by just gathering that information. Processing conditions, slip agents, mold release agents, what happens to sterilization to the material or to those materials, and intended use, of course, is important here. Regulatory status of the material, where has it been used? This is kind of our historic literature search that we've done in the past. What do we have to our advantage in potentially competitor products on the market, or materials, that is, that may be a little bit harder justification, because I think we all know there is tons of adulteration that happens to our materials in the processing steps of creating our devices. However, that information could always support and be welcome to support. And then step 2 would be conducting that chemical characterization testing. So we have our extractables or leachables testing, right, to understand potential migrants from our materials, and also our expected leachables under expected use cases, and potentially in simulated use cases as well, as we see in leachables testing. There we're identifying our organic compounds, or inorganics, or our heavy metals, as mentioned, and in doing so, our volatile and semi-volatile substances, typically. There we're using multiple analytical methods, as we normally would during those chemical characterization tests. Then we move into step 3, identifying and classifying those chemicals. So we would match those detected compounds to known chemicals, right? We do this by, or compounds. We typically do this by using spectral libraries, different reference standards and testing, literature data, expert analysis in the laboratory, and also proprietary databases as well. And so we classify these chemicals and their known toxicological profiles, and we understand how we classify those per our relevance of the intended use. From there, we'd move to quantifying exposure. We determine, worst case daily exposure as an example, based on the highest measured concentration of those extractable leachables, and we normalize those to our patient doses and through that toxicological risk understanding. So in doing so, we're comparing the safety thresholds. So we're using toxicological databases. This process would be done by a well-qualified assessor to be able to help here and be able to identify potential toxicological risks. And then when specific data is unavailable, we are moving into other forms of risk mitigation, such as applying our TTC values or Kramer classifications, et cetera. There we'd move to risk categorization. So with all that information, we would compare our worst daily exposures to a potential safety threshold, and we would move into determination of effects of different toxicological endpoints. And from there, we would wrap up our decision and documentation through the remainder of our safety assessment here. We would summarize if any compounds are below safety thresholds, or if any exceed safety thresholds. Again, the certified and appropriate evaluator assessor would make a risk estimation, and we would document that and justify for our safety conclusion. So that's a little bit information about, or a little bit of a breakdown about our chemical safety assessments and the steps that should go into those. So really, as we conclude, this is an industry call to action, right? These recent changes to USB are very exciting. It's great to further harmonize with ISO 10993, especially with the dramatic changes to ISO 10993. And if you are unaware of those changes, just a quick plug for Nelson Labs as well. You could review our webinars there on those changes. In vitro testing has been combined with that more risk-based approach. And so we're being more aware of part 2 of ISO 10993, where we want to focus on animal welfare. We want to provide better science that is more specific and more sensitive to our use. And we know that the industry must drive this adoption. So in summary, in vitro methods that we are seeing now move into these changes of USP are here to improve ethics, science, and efficiencies. And Nelson Labs is committed to an animal-free future. So, we do hope that manufacturers do see the benefits here, and we'll look forward to these changes moving forward. So again, a last reminder: these changes do go into effect in December of 2026. However, the USP has published for, again, that early adoption now. So if you are considering that testing for materials or working with your suppliers and understanding the status of their materials, I would be recommending that you refer to these revisions as you gather that information. So thank you so much for listening and attending. I'll pass it over, I believe, back to Omar for our Q&A session.
And now on to the question and answer portion of our event. For this segment, we welcome Audrey Turley, our presenter from yesterday's session, to assist in answering any questions you have today. To participate in the Q&A, just type your question in the text box located to the right of the presentation window, or click the Q&A icon at the bottom of your screen. If we're not able to answer all submitted questions during today's webinar, we'll be sure to share them with our speakers, who can reply to you offline. And our first question is, there's a persistent interpretation that animal testing is a mandatory step for all medical device approvals in the US. What's your take on this?
Yeah, so I'd be happy to start there. And if Audrey, welcome your feedback too here. So that is certainly accurate. The persistence in the US for animal testing is very real. I think what we have seen is kind of just a quick summary, right? And a reminder is that I do believe the EU has paved the way. The cosmetic industry, again, driven by consumers and their feedback, has really paved the way to move towards more sustainable models in these in vitro models. Unfortunately, the US has always loved their animal testing. Now, we have been starting to make good efforts with recognition of these changes to in vitro models and their availability. However, there is a risk, right? There's a real risk for a medical device manufacturer conducting this test and for submission to the US and having a regulator respond to say, hey, I still want to see the animal testing. I will say we're starting to see more and more manufacturers leverage the in vitro model and more acceptance of that in vitro model. But you're accurate. You're right in that the U.S. is pretty much I'd almost say last to the table here in the acceptance of these animal models. So we do want to continue to push for this testing. What is nice here is our conversation today is around USP testing, so materials. Right. I do want to remind that our finished final goods should still be subject to ISO 10993 review, where that may change decision a little bit more. And then also, as I mentioned, our position globally and where we are submitting for regulatory approval. Audrey, I don't know if you have anything to add to that as well.
Yeah, I would just add that, we need to consider the evolution of science that has occurred over the last several decades. And when we started to do these biocompatibility or biological safety assessments of products, the science that we had at the time was still from the 50s and 40s, 1940, 1950, and we're actually still leaning on that because we have a lot of history that it has proven to protect patient safety and proven to predict issues. Now, not all the time and not for everything. But what we struggle with now is, what's an appropriate validation of an in vitro alternative method? So we have a lot of discussions about that. And as we move forward, the in vitro irritation was one of the first ones we looked at replacing just recently. I say recently loosely, because that was, like 11, 12 years ago. And we're still working on that. But as we move towards sensitization, we're looking at how do we have a proper validation. And also, it's not just about numbers and statistics, it's also about compounds and chemicals that we look at, what's used, how we're formulating new compounds and chemicals and materials, and how we want to assess those as well. So we can look out for the unknowns, that we're discovering actually do cause quite a bit of harm. How can we predict that up front? And so while the US is very cautious, it is from a scientific perspective. And they, we're having a lot of conversations about how to navigate that road and make sure that patients are still safe.
Next question is, why was NRU included into USP 87 and not MTT or XTT?
Yeah, I can take that, Nick, if you want me to. So it's more based on familiarity. So in the pharmaceutical world, which is what USP is focused on, NRU is a more comfortable assay with more familiarity. So it's really just based on what the industry is comfortable with. Not that there's no awareness of MTT or XTT, and the decision came down to what was more broadly available.
And our next question is, what is the understanding that, I'm sorry, let me restart this. So this question comes in the form of, apologies, I am not that familiar with the testing of devices. It was my understanding that medical devices follow the ISO framework. In which situations is the USP framework used? What is the difference in regulatory acceptance between ISO and USP?
Yeah, so that's a great question. So really, what we found historically is a lot of USP testing. This really started, again, if we kind of recall, from understanding our drug products and quality of drug products. And so those pharmaceutical packaging products were really brought into play and considered. As industry has grown, we've found that medical device industry is often leveraging a lot of the same base components, plastic, polymeric materials, as some pharmaceutical packaging systems and delivery systems are using. And so it's become a little bit more widely accepted to consider USP testing to support your biological evaluation towards your finished final good of your medical device. Now, it is accurate that the finished final medical device is subject to the overview of ISO 10993 and those regulatory standards within and the chapters and sections within that standard. But that USP testing and their testing of materials could be used to further support those device evaluations. The one caveat I want to make sure to highlight there is that conducting USP testing for a medical device does not consider the medical device biocompatible and risk-free, right? Or acceptable limits of risk, right? The testing from USP, the USP guidance in that testing, those test methodologies may overlap with ISO 10993 testing, but they are to support the use of appropriate materials in both pharmaceutical packaging systems, delivery systems, and also, as we're mentioning now, often widely used in medical device applications. So one thing to keep in mind is that we often adulterate our materials downstream, right? We may bring in materials and into our manufacturing processes, we may coat them, we may cut them, we will clean them and sterilize them, et cetera. And now we are dramatically adulterating the potential for biological or biocompatibility risks. And so therefore, that is the reason why ISO 10993 is then our governing umbrella document over those finished final medical devices. Hopefully that made some sense. Audrey, feel free to add any if need be.
No, I think that was great.
Okay. Hopping to our next question, when would you use USP versus ISO 10993?
I'd say that it's pretty similar, so without having to spend too much more time on that. Again, consideration of material changes. So if I'm a medical device manufacturer and I have material suppliers, I may like to see USP, testing or adherence to USP testing from my material suppliers, so that I have a greater level of confidence in the material I'm bringing into my facilities, and knowing that I will have a lower likelihood of downstream risk associated with biological safety of my finished final good. So that being said, if I have material supplier changes, there may be changes in manufacturing fluids of that raw polymer bar stock, right? Or there may be a base polymer change where they're sourcing those polymers. Potentially the material comes in white and now it comes in red and there's a supplier change. I may want to consider that testing associated with the USP chapters that we reviewed today to support that material coming into my facility and knowing its pedigree, if you will.
Do the new changes apply to the packaging materials?
Yeah, so this is a good question.
Go ahead.
Sorry.
Go ahead, Nick.
So it does. There are some nuances though. So the test for in vivo, we're removing and creating a little bit more push towards in vitro testing. So after the change here, packaging materials can no longer rely on that Class 6 proof of safety, if you will, and so they are subject to very similar tests, right? So these materials that come in contact with our drugs, devices, or biological systems, including those container enclosures and packaging, would be considered within the scope of Chapter 87 for those in vitro approaches as well. And so some of that guidance is also highlighted within the informative chapter, Chapter 1031 that we discussed as well, which is very, if I will, very packaging focused.
We have time for about one more question. And the next is, are the changes acceptable by regulatory bodies globally? And will these changes vary for raw material suppliers versus final product manufacturers?
Yeah, that's a great question. So in terms of product manufacturers and material suppliers, I would be holding my material suppliers, accountable to these changes and to these published changes for these upcoming revisions that, again, will go live in 2026, December of 2026, but available now for review and leverage. Now, global acceptance of USP. Yeah, I don't know, Audrey, if there's anything else you want to add to, I guess, how that is viewed.
Yeah, I think that this could be a mixed question as we move from in vivo to in vitro. What's the global acceptance there? And I think it's important that we keep, some guardrails around that conversation. So when we talk about screening materials for med devices, there isn't a criteria to meet. It's really being thoughtful about the customers that you market to. So you can follow USP. You're welcome to still follow 10993. That's probably more of my favorite for med device manufacturers and raw material suppliers to really focus on 10993 applications. Because there are differences in the USP testing that is performed. And then when your customers wonder, is that data relatable to the final evaluation of my device? We get asked that question a lot. And sometimes we really have to dive into that and understand, oh, you did this, but you missed one portion. So we really have to look at it differently. So if you are a raw material supplier for med device companies, I fully urge you to look at 10993. If you supply to pharma and med device, then I say USP 6 is really a good place to start. Every country has its own pharmacopoeia that they follow. So USP, we have an EP, a JP, so there's different ones around the globe. What's important, and I'm not able to speak to the regulatory acceptance there, but what is important is to know the expectations from the different pharmacopoeial guidelines that are published in the countries you are submitting to. So if you're manufacturing in the US, clearly USP is something that you'll be familiar with. But again, thinking about where you're distributing to, if those will be lined up correctly. And I can't speak to whether the Japanese or European, those are the ones I'm more familiar looking at, but I don't know if they've updated their chapters to match.
Yeah, and similarly, just to add to that, the concept here is acceptance of these USP changes is also in alignment with the understanding of the acceptance of the shift to in vitro models, right? As we've realigned or aligned these changes and these revisions to these chapters to be more in focus with also ISO 10993, aside from just being more animal free and more in vitro focused, that also will be into consideration with the market in which you are looking to submit for regulatory approval and review as well. And so we talked a little bit about the U.S. being behind the eight ball, and in terms of acceptance of in vitro models, we are making good progress, but the EU is certainly leading that charge. I will say Asia may require a little bit more time from my personal experience. However, they have recognized the in vitro irritation model. So that is good, and carries a good amount of weight as well in that. So as we continue to shift as an industry to using these models, we hope for further and more widely accepted feedback.
Awesome. Well, that's all the time we have for questions today. Thank you, and we appreciate your time and expertise on today's topic. Thank you to our sponsor, Nelson Labs, as well as to everyone in the audience. We appreciate your attention and participation. Within the next few hours, you will receive a personalized follow-up email with details and a link to today's presentation on demand. Please feel free to invite your colleagues and peers who may not have been available to listen to the event. On behalf of our guests, Nicholas Cristiano and Audrey Turley, I am Omar Ford. We look forward to seeing you again tomorrow.